to me when you're ready, and I'll introduce you.
Okay, just 1 more minute. Yeah, still a couple of people who are not here, but they can file them later. Yeah.
So welcome everyone. It's a great pleasure to have Kyle defending his thesis today. Kyle graduated with a Bachelor's of Science in Chemistry from the College of Worcester in 2018, and then joined us here that fall and was in the thick of things in terms of COVID, and we survived that, and he's made it to this date. He's going to share with us some of his graduate thesis work that involved understanding the mechanistic impact or roles of nitric oxide on some of the antibacterial effects that we study. And so with that, Kyle.
Thank you, Mark, for that introduction, and I'm very excited to share with everyone today some of the work that I've been doing for my PhD. Okay. Nowadays, I think we hear a lot about antibiotic resistance and how it's a big problem in medicine. And it is a big focus in my talk, so I first want to introduce this concept. So when there is an infection, there is a bacterial population that we want to eradicate. And when given the antibiotics, then most of the bacteria that are susceptible to the drug, shown here in blue, will be killed. But due to random mutations, there is a small number of bacteria that are resistant to the antibiotics and can survive. And after the subsequent treatment, then all of the susceptible bacteria are gone, but the resistant bacteria can survive and propagate. And at the end here, we have a population that is entirely composed of resistant bacteria. Now, antibiotics were first discovered in 1928 with the isolation of penicillin. And in the following decades, there was a boom in discovery of different antibiotics classes. We have beta-lactams and glycopeptides that inhibit cell wall synthesis, aminoglycosides, anthomycins and cephalogrammins that inhibit protein synthesis, and quinolones that disrupt DNA replication. However, since the 1980s, there was a big decline in the discovery rate, and since 1990, there has been a void in antibiotic discovery. Now, couple that with the overprescription of antibiotics in clinical use, there is a rise in antibiotic-resistant infections. As an example, shown here in this graph, we have 3 different pathogens: Staph aureus, Pseudomonas aeruginosa, and Enterococci being resistant to different types of antibiotics. And as I've listed here, different classes of antibiotics have different mechanisms, and bacteria can also develop different ways to survive, different strategies to develop resistance against these antibiotics. Now, generally, to be effective, the antibiotics have to traverse the bacterial membrane and target a protein inside the bacterial cell to inactivate it. So the bacteria can change the target, protein so that the antibiotic cannot bind to the drug, then it cannot be effective. Bacteria can also produce enzymes that degrade the antibiotics before they can be effective. They can modify the structures of their own membrane so that the antibiotics cannot go inside the cytoplasm. And once the drugs get inside the cytoplasm, there are also efflux pumps that can remove the drugs from the cytoplasm. Now, these mechanisms apply to only one bacterial cell, but bacteria can also attach and grow into these dense aggregates, and they secrete a polymeric matrix. And in this form, it's called a bacterial biofilm, and bacteria in this way, they're basically embedded in an environment that provides them with additional protection against antibiotics. For example, the dense aggregates of the biofilm means that bacteria in the core regions of the biofilm experience lower oxygen and nutrient conditions than bacteria on the surface of the biofilm. And this is damaging to the antibiotics because the bacteria in the core regions that will grow more slowly, and antibiotics require active growth to be effective. Because bacteria are very closely packed inside the biofilm, they can also exhibit quorum sensing, meaning they can secrete signaling molecules that allow them to alter their gene expression and different physiological processes. Now, this communication allows this bacterial community to survive collectively. The extracellular matrix of a biofilm contains like a sticky polymeric substance that contains DNA, sugars, and proteins, and these are overall negatively charged, so they can bind to the positively charged antimicrobials before they can reach the bacterial cells. The matrix is also viscoelastic in nature, so that means they can drastically slow down the diffusion of the drugs. And finally, many biofilms that we find clinically are polymicrobial in nature, meaning there are multiple species coexisting in the same biofilms. And this allows them to exchange genetic material and gaining resistance in that way. Because like one species can have the resistant genes that get passed on to a different species. So because of these mechanisms that I have explained, as well as the rise in resistant infections, there is a need for a novel antimicrobial agent that does not confer resistance. And nitric oxide is one such promising agent. Nitric oxide, or NO, is a endogenously produced free radical, and it has many different functions in the human body, but I want to focus on its role as a pathogen killer here. And NO is good at killing bacteria because of its ability to generate reactive molecules. Outside of the cell, NO can react with oxygen to form NO2 and eventually N2O3, which can nitrosate protein residues that have thiols, such as cysteine. And this will alter the protein structure as well as its function. NO can also diffuse inside the cytoplasm of the bacteria, and here it can react with superoxide to form peroxynitrite, causing lipid peroxidation, and as well as DNA damage. And this provides NO with a critical advantage over antibiotics, because antibiotics usually only have one mechanism of action for each drug, whereas NO has several, and so it's harder for bacteria to gain resistance against NO because they would have to develop multiple mutations at once. Now, nitric oxide is normally a gas, and it is difficult to administer a gas to treat an infection because it requires complex equipment, and you need a very high concentrations of NO in gaseous form that might lead to toxic side effects. So to solve this issue, we can use solution phase nitric oxide donors to chemically store NO and then release it in media to sustainably deliver a dose of nitric oxide. And in one of the more prominent types of this chemistry is using diazinium dilates, where we can subject a secondary amine to high pressure of NO gas under basic condition to have this moiety here, and diazinium dilate, which can then be decomposed in a proton-initiated manner to return the parent amine with 2 moles of NO. And small molecules have been developed to utilize this chemistry to release NO, but there are also macromolecular systems that can utilize this moiety. For example, we can load small molecule NO donors inside liposomes to better target phospholipid membranes. We can use dendrimers with a positive charge to increase toxicity to bacteria. We can also use biopolymers such as oligosaccharides to improve cytocompatibility. And for my thesis, I've been I've been trying to study different aspects of the interaction between the NO and bacteria beyond just learning about the killing potential of these materials. And so today I'll be talking about chapters 2 and 3. And first of all, chapter 2. Chapter 2 aimed to examine the different effects of NO on two major bacterial classes, gram-negative and gram-positive bacteria. Gram-negative bacteria is characterized by having an outer membrane as well as a cytoplasmic membrane sandwiching a thin layer of peptidoglycan. And peptidoglycan is this network of peptide sugars that provides rigidity to bacterial cells. And for gram-negative bacteria, this layer is very thin, it's only a few nanometers in thickness. For gram-positive bacteria, they only have one layer of cytoplasmic membrane, but instead they have a very thick layer of peptidoglycan, about 30 to 100 nanometers. Now these key differences result in different interactions between antibiotics and the bacteria. Usually antibiotics are less effective against gram-negative bacteria due to the double layer membrane. And so I hypothesized that due to this difference, NO may also have different effects on the two gram classes. And so the goal of this project was to examine the impact of NO on the morphology of these two gram classes. In this study, I selected pathogens from the escape pathogen group, 2 gram-positive and 2 gram-negative bacteria. And these escape pathogens are known to have high incidence of antibiotic resistance. For this study, I predominantly utilized microscopic techniques such as transmission electron microscopy and atomic force microscopy to answer my questions. So in our lab, we use a system that you see on the left here as an automated system, and they are basically steel chambers that can withstand high pressures of NO gas that we can put our secondary amines in and then subject them to 20 bars of NO for 72 hours and with a basic catalyst, sodium methoxide, and then I extract and wash them with cold diethyl ether to yield the final product. And for this project, I selected 4 NO donors. 4 NO donors: Sperno, Papa NO, DPTA NO, which are small molecule NO donors with different release kinetics that I'll be talking about in the next slide. I also selected NO releasing beta cyclodextrin, or CDNO, to represent an oligosaccharide NO donor. And so after the NO donors were synthesized, I used a technique called a nitric oxide analyzer to quantify the NO release. And this technique involves dissolving a small amount of NO donor in a flask of PBS, which will then liberate NO. And the NO will then be carried by nitrogen into a reaction cell, where it can react with ozone to form an excited state of NO2, which can then, when it relaxes down to the ground state, can liberate a photon that can be amplified with a PMT and detected by a computer. Now, using this technique, we can measure in real time the NO concentration and determine, for example, the maximum instantaneous NO concentration. And if we integrate this graph, we have the cumulative NO release over time, where we can also see the NO totals as well as the half-life of the NO release. And for the NO analysis of these NO donors, I found that Papa NO, Sperno, and DPT NO have higher NO payloads compared to CD NO, and this is not surprising because these are small molecules compared to the oligosaccharides in CD. And also, Papa NO has relatively short half-life, 0.4 hours, and Sperno and DPATA NO had progressively longer half-lives, meaning they have a more extended release profiles. CDNO has half-life of about 1 hour. Okay. So first I wanted to look at how nitric oxide will affect the bacterial membrane. And to do this, I use transmission electron microscopy, or TEM, to visualize the cross-section of the bacterial cells. So very briefly, in this technique, we have an electron beam that passes through a series of lenses to hit a specimen, and the transmitted electrons then go through more lenses and are finally captured by a detector as an image. Now, this method provides very high resolution, and therefore can be used to visualize the bacterial membrane, but it requires very thin specimens so that the electron beam can pass through. And so for the sample prep, I first treat the bacteria at lethal doses of NO. Then I fix the bacterial cultures with glutaraldehyde to cross-link any proteins and preserve them from subsequent sample prep. I stain the bacteria with a metal dye, osmium tetroxide, dehydrate them with ethanol, and then embed them in epoxy before cutting using a knife, a diamond knife, to cut very thin slices, about 60 nanometer slices. For reference, bacteria is about 1 order of magnitude thicker, 1 to 3 microns. Okay. So first, we have untreated gram-negative bacteria shown here, Klebsiella pneumoniae and Pseudomonas aeruginosa. You can see that they have intact cell membranes with no spillage or disrupted membranes. When I added Spurno and CDNO, you can see a heavy disruption of the bacterial cell envelope. The bacteria lose their shape, and there's some spillage of the cytoplasmic content. Now, for gram-positive bacteria, Staph aureus and Enterococcus faecium, untreated, you also see like an intact membrane, and you also can tell that the membranes of these bacteria are thicker, visibly thicker than the gram-negative, and this is due to the thick peptidoglycan layer. But what I found was interesting was that when I added NO treatment, there was no visible changes to the bacterial cell envelope. And I went further and tested this with see if different NO release kinetics will have the same effect. And what I found was that regardless of the NO kinetics, Papa NO being the fastest and DPTA NO being the most extended release, Klebsiella pneumoniae, so gram-negative bacteria, had their membranes disrupted, whereas Staph aureus, the bacterial cell envelopes were not affected. I also tested with a few drug-resistant strains and saw the same result. Again, gram-negative was affected, and Staph and gram-positive was not affected. So, regardless of the treatment condition, it appeared that the gram designation was the main factor that led to the bacterial cell effects. So that was looking at the cross-section of bacteria. Now I want to look at the sort of the exterior of the bacterial cell, and to see how, specifically to measure the surface roughness. And this is important because increased surface roughness is usually correlated with increased membrane damage. And to do this, I use atomic force microscopy, or AFM. And how this works is we have a nanometer size metal probe with a laser shining on top of it that can be detected by a photodiode. And as the probe lowers onto the sample, it will bend and get deflected, and this deflection can be measured by the laser spot to produce an image. For the sample prep, I first start with a layer of mica, which is a silicate material that is atomically flat. I then add a layer of polylysine to provide a positive charged surface that can then be used to attach the bacteria that has a negative charge on the outside. I then treat the bacteria in this manner by submerging it in media, and then dry off the excess liquid to begin my imaging. And this is an example of what an image would look like. You can see bacterial cells sitting on the surface. and I zoom onto one bacterial cell surface, and I draw a red line across the surface, and this will trace the surface roughness of the bacteria. And after doing some flattening and applying an equation, I can then determine the RMS, or the root mean square roughness of the bacterial cell. And on the left side here, you see Klebsiella pneumoniae gram-negative bacteria having higher RMS roughness compared to untreated bacteria, and also compared to non-NO-releasing analogs in the hollow bars. For Staph aureus, the gram-positive, I did not see any such change. Again, agreeing with the TEM images. And I also tested this with using different NO release kinetics, Papa NO, Spur NO, and PTA NO, and again, I saw the same result, where all three NO donors appeared to increase the RMS roughness for gram-negative bacteria, but not for gram-positive bacteria. So again, NO treatment only seems to affect the gram-negative bacteria, but not gram-positive bacteria. And at this point, I wanted to check if it really is that the bacteria's envelope of gram-positive are not affected at all by NO treatment. And to do this, I utilize a fluorescent technique using this fluorophore DISC35, see here as a blue squares. And how this works is that if there's a healthy bacterial membrane, then during hyperpolarization, the dye can translocate onto the membrane of the bacteria. This is a gram-positive bacteria, so you would have a low fluorescent signal. If the membrane is compromised, however, it cannot retain this fluorescent dye, and so the dye will stay in the media, and you have high fluorescent intensity. This is an example of data. I would wait 5 minutes for the baseline to stabilize before adding the fluorophore at a time of 5 minutes. And if there's just media and no bacteria in the well, then you have a spike in fluorescent signal and a slow decay. And this decay is due to the dye sticking to the polycarbonate surface of the well plate. If there is untreated bacteria in the well, then you can see that this spike in fluorescence quickly goes down to like a really low steady state. And this is due to this intercalation of the dye onto the membrane. When I added my NO donor, the example here is spermino, the fluorescent becomes high again, meaning that signifying that the cytoplasmic membrane is compromised and the dye stays in the media. And as a control, I also included rifampicin, which is an antibiotic that interferes with DNA replication. And this is not a membrane active process, so the membrane stays intact and you have low fluorescence. So this showing that this indicates that the cytoplasmic membrane of gram-positive bacteria are disrupted during NO treatment, but the rigidity and the cell shape was retained by the peptidoglycan layer here. So in summary, again, gram-negative and gram-positive bacteria, they differ in their membrane makeup. And I use TEM and AFM to determine that gram-negative bacteria, the envelopes were disrupted, as shown by the puncturing of the membranes as well as increased RMS roughness. Whereas gram-positive bacteria, the membrane is also compromised, but the thick peptidoglycan layer retains the cell shape. So for this, that chapter, I investigated 4 bacterial species. And for the next chapter, I focus on only one species, Klebsiella pneumoniae. And I want to talk a little bit more about antibiotics because in the introduction, I said that there are many different types of antibiotics and they differ in mechanism of action, but they also differ in how much they're used. You can see in this pie chart here, this one class beta-lactam accounts for more than half of all antibiotics in commercial use, and this is because beta-lactams are better tolerated by eukaryotic cells, so they produce fewer toxic effects. And Klebsiella pneumoniae is a gram-negative bacteria. It is ubiquitous in the environment, and it is dangerous because it can cause infections and produce enzymes that are effective against beta-lactams. And this is problematic because then you have to resort to using other antibiotics such as colistin that have really bad side effects. So Klebsiella is broadly classified into two groups, classical and hypervirulent strains, and they differ in some key area. Classical Klebsiella can cause UTIs, bladder infections, and pneumonia, whereas hypervirulent strains usually they're associated with more severe diseases such as pyogenic liver abscess or meningitis. Classical strains are hospital acquired, meaning they often affect people with a weak immune system, whereas hypervirulent strains are community acquired, so they can also affect healthy hosts. Classical strains are overall considered to have higher rates of resistance compared to hypervirulent strains, even though more recent reports have also reported increased incidence of resistance for hypervirulence. And I just wanted to show the headline of an article in 2016 of a patient who got a Klebsiella infection after traveling, and this particular strain was resistant to all available antibiotics, and the patient succumbed to the infection, just to show how deadly this bacteria can be. And to dive a bit deeper, Klebsiella has 4 main virulent factors. And virulent factors are just characteristics of their physiology that make a patient sick. Klebsiella has lipopolysaccharides, or LPS, which is a component of the outer membrane of gram-negative bacteria, and they can defend the bacteria. They can protect the bacteria from humoral defense, as well as recruit inflammatory cells. Klebsiella also has siderophores, which are small molecules that can sequester iron from the host, and this is important to keep the bacteria alive. Klebsiella also uniquely has a layer of capsule, which is a polysaccharide matrix that basically masks the bacterial cells from the immune cells, such as macrophages, so they don't get recognized. And they also have these fimbriae on the surface that allow them to attach to different substrates and form biofilms that I can touch on later. And so the goal of this project was to examine different aspects of Klebsiella physiology when subject to NO treatment, and namely resistance profiles, growth inhibition, the capsule characteristics, as well as some biofilm dispersal work. For this study, I chose 4 strains of Klebsiella. MKP, MGH are classical strains, and KPB-R1S and NTUH K2044 are hypervirulent strains, given how different the two classes are, and they can react differently with NO. I also selected 4 NO donors, so Papa, NO, Spermino, and DPTA NO that you've seen, as well as MD3, which is another small molecule NO donor that has similar NO release kinetics as DPTA NO, 5.1 hours half-life, so more on the extended side. I also chose meropenem, which is a beta-lactam antibiotic, as a positive control. So the first thing that I wanted to do was to determine what concentrations of these NO donors were needed to kill the bacteria. And I did this using a planktonic inhibitory assay. And how this works was that we have these wells here, each containing bacterial culture, and they're seeded at very low density so that the media remains clear. And in each row, I would add a drug with descending concentration across the row, so 400, 200, 100, and so on. Now, and then I'll incubate the well plate for 24 hours at 37 Celsius. And what happens after the incubation period is, for some wells, the drug concentration is low enough that the bacteria can grow in density and become turbid, shown here in orange. But as you increase the concentration, it comes to a point where the concentration of the drug is high enough that it inhibits the bacterial growth. And the first well where that happens is considered the minimum inhibitory concentration, or MIC, and that will be reported for each drug. And what I found from this assay was that the NO donors, Papa NO, Sperno, PTA NO, and MD3 had similar MICs between classical and hypervirulent strains. And in each of the rows, you can see that they differ only about maximum by a factor of 2, which on this schematic would represent 2 neighboring wells. And I also found that meropenem was able to inhibit bacterial growth at much lower concentration. And this is good, but we know that antibiotics can develop resistance. Bacteria can develop resistance against antibiotics, and that's what I wanted to study in the next using serial passaging experiments. Now, in this experiment, I first start with an MIC experiment like the same one as we showed in the previous slide. And you can see the MIC here shown in green. I would then go one row below that where the bacteria grow. So in this case, we have the MIC, and use this bacteria to seed the next culture and do the same experiment again. So we have a new MIC for passage 2, passage 3, and so on. And if resistance developed, then what we would find would be an increase in MIC as the passages just go on. So from 25 to 100 to 400. And for meropenem, the 2 classical strains, NKP and MGH, develop resistance from passage 1 all the way to 14, with the MIC at pass 14 being roughly 500 times higher than that at 1. The hypervirulent strains, NTUH and KBBR1S, did not exhibit this resistance, and that's not surprising given the prior knowledge. For Papa NO and MD3, 2 NO donors, we did not see resistance developed for any of the strains, showing that NO, due to its multi-mechanistic killing, was able to prevent the bacteria from acquiring resistance. I also wanted to look at how, examine whether NO inhibited the growth of Klebsiella culture. And for these experiments, Klebsiella were treated with sublethal doses of NO and monitored using, monitored with optical density to see how fast they grow. First of all, in the trays in black, you see that control cultures start growing right away. After the half an hour, you can start to see an increase in optical density. And meropenem line in red closely resembles that of the control, showing that meropenem did not inhibit growth. MD3 inhibit growth a little bit, and that we think this is due to the low NO dose that was given at this concentration. We also found that Papa NO, Spermino, and DPTA NO had all partially inhibit the growth of Klebsiella to different extents. Papa NO, the bacteria grew at around 6 hours, and progressively Sperno and DPTA could inhibit bacteria for longer. And I noticed that this trended with the release kinetics of these NO donors. Papa NO had the fastest NO, the shortest NO half-life of 0.7 hours, followed by Sperno and DPTA NO. And represented here is the instantaneous NO concentration with time. And you can see that Papa NO in green, it releases most of its NO in the first few hours. But in the inset, you see that after 3 hours, the level NO in Papa NO decreases below that of Sperno in orange and DPT NO in blue. And this indicates that there is a threshold level of NO needed to maintain, to prevent NO bacteria from growing. And Sperno and DPT NO were able to maintain this level for longer. Okay. Next, I examined the capsule production of these Klebsiella bacteria, and I do this by measuring the level of uronic acid, which is a component of the capsule. And for this assay, I first start with an NO-treated culture. Then I add a surfactant to dissolve the uronic the capsule and separate it from the bacterial cell. Then use an aliquot to precipitate in cold ethanol, dissolve it in sodium tetraborate in concentrated sulfuric acid, and then add 1-phenylphenol to turn the solution red, which can be detected on a plate reader. First of all, we found that classical Klebsiella pneumoniae on the left had lower capsule production compared to uronic acid levels compared to the hypervirulence on the right, and that's not surprising because hypervirulent strains are known to produce higher level capsule, but I did not see any statistical significance in any of the treatment conditions, showing that NO does not affect capsule production. But there's another property of capsule, which is mucoviscosity, and this has to do with the gooiness or the stickiness of the capsule layer. And we can visualize that by pipetting the bacterial culture in a cuvette. For a hypervirulent strain, for a hypermucoviscous strain, you can see that the capsule is kind of gooey and it forms a string like that in appearance. But for a similar strain that produces the same amount of capsule, but it's not mucoviscous, then you can see this jet that's not gooey. And this property is important because some hypervirulent strains, hypermucoviscosity, so the gooiness is more highly correlated with hypervirulence than simply just the capsule production. Now, we still found that classical strains have lower supernatant, lower mucoviscosity compared to hypervirulent strains. But what I found was that for hypervirulent strains, NO donors, and notably Papa NO, Sperno, and DPT NO all led to a decrease in mucoviscosity, showing that these NO donors were able to reduce capsule viscosity. And finally, I examined NO interactions with Klebsiella biofilms. Klebsiella biofilms can be a problem because in this one study, collecting Klebsiella cultures from clinical isolates, about half of the culture shown in the red box were able to form biofilms. And as we know, that biofilms can develop more mechanisms to have resistance against antibiotics, and these biofilms are usually associated with the medical devices like catheters. You put catheters in the patient, they can develop a biofilm infection. Now, for this study, I grew biofilms by incubating a culture of Klebsiella in M63 minimal media, supplemented with glucose, for 24 hours. After that, I rinse off the excess liquid, and what I have is a layer of adherent biofilms stuck on the bottom of the well, which can be visualized using crystal violet. You can see at the bottom, a very deep purple color would mean there's a lot of biofilm formed. And I tested the baseline experiments and found that of the 4 strains, MGH, the MGH strain, the classical strain, formed the most biofilm. So I selected this strain for further dispersal experiments. Now, for this data, the x-axis corresponds to concentrations, whereas the purple line corresponds to biofilm mass, and the black corresponds to viability, so how many bacteria are alive in the culture. And for meropenem, as the concentration increases, we saw a decrease in biofilm mass, signifying there's a dispersal. But the viability only decreased by 1 log, all the way up to about 100,000 times higher than the MIC, all the way on the right side, showing that even though meropenem could disperse the biofilm, the bacteria stay alive and are insensitive to the antibiotic. Now, the 4 NO donors, MB3, Papa NO, Sperno, and DPTA NO, all showed both a dispersal of biofilm in the purple trace, as well as a significant drop in bacterial viability in black, up to 5 logs. And this shows that NO donors can both disperse as well as eradicate the bacterial cells in the biofilm. And I also visualized this using scanning electron microscopy. For control, the control sample, you can see these dense bacterial aggregates, which is indicative of the presence of biofilm. When meropenem is added, you can see there's some thinning down of the bacterial aggregate, but there is still a lot of cells on the surface. For Papa NO, Sperno, and DPTA NO, you can see a virtual a significant reduction in the aggregate with only a few cells left. And an interesting case MB3 was, even though I verified that these cells that you see are dead, that you can still see a layer of cells stuck on the surface, and we are hypothesizing that that's due to the polycationic nature of MB3. It has multiple sodiums in its structure that can provide a positive charge that sticks the bacteria to the surface. But overall, this shows that NO donors can disperse the biofilm as well as killing the bacteria within it. So, for conclusion, for chapter 3, Klebsiella again can cause very deadly and highly resistant infections, and I showed that Klebsiella did not develop resistance to NO. NO can partially inhibit Klebsiella in a release kinetics matter. NO treatment does not alter capsule production, but it does reduce the mucal viscosity, and NO can also disperses and eradicates Klebsiella biofilm. For future directions, a metabolomic analysis of exogenous NO and 2 bacteria would be useful to elucidate further interaction between NO and the bacterial cells. Metabolites are small molecules that are produced by bacteria or cells, and they reflect the overall health, physical state of the bacteria, and as well as different metabolic pathways happening. And metabolomic knowledge of exogenous NO on pathogen is largely unknown. I'll just highlight one example of a study that I found, which in this study they treated a gut microbiota, with an NO donor, and they found that over time, after the treatment, there is an increase in leucine, which signifies a shift in amino acid metabolism, and a decrease in indopropionate, which signifies an anti-inflammatory marker. It's a typical workflow of metabolomic analysis. Is we start with the bacterial culture, which can be untreated or treated with NO. Centrifuge the culture so that you can separate out the supernatant and the pellet, the bacterial pellet. The supernatant can be used to analyze the metabolites that the bacteria secrete into the media, whereas the pellet can be used to analyze the intracellular metabolites. And then you can do physical or chemical extraction methods, as well as pre-concentration to improve sensitivity, and to have a sample that can be and data collection can be done using liquid chromatography coupled with mass spectrometry before analysis of the data set. And with that, I would like to thank my committee members. First, I want to thank Mark for giving me an opportunity to do research with you. Mark is a very demanding advisor, but he also showed me a lot of grace in when times are hard. I also want to thank my collaborators, Dr. David Hill and Kim. David helped me a lot with the mucus experiments for my project, and Kim is wonderful with my Klebsiella project. Thank you. Gary and Leslie, thank you so much for being here. They're not only on my committee members, but I've also got a lot of help from other members of the lab, like Tyler and Cam in your case, and the whole Higgs lab. I've asked them for a lot of help too. And my collaborators and the Hill lab, the Kessimer lab, King, Miller, and Wallet labs, all have contributed to my project. Brandy from the Mass Spec Core, Brian and Keith are two AFM experts that helped me a lot with the AFM studies, and Amar from Channel helped me a lot with electron microscopy experiments. I also wanted to thank No Bio for the funding support, as well as the core facilities in Channel, Bioinformatics Core, and Mass Spec Core Critical, as well as past and present members of the Shanefish Lab. All of everyone who I've had an interaction with has helped me along the way. Their name is on here. I won't say out all of your names, but just want to let you know I appreciate each and every one of you. And just some photos. I had a lot of fun in the Shanefish Lab. We have, bring up the laser pointer. Yeah, this is the dodgeball tournament that I think we've got one win, right? One win, that was fun. This is Mark, whose height is being measured by Quincy. We have a height wall in the lab. This is the lab just doing like a plant infection study, and just other silly pictures of me trying to fix the GPC, and apparently also trying to fit into a box. And my family and friends are also here. We have my friends from Singapore who are either here in person or watching online. Some golf bros who have spent a bit of money on golf over the years, so it was really fun. My family and friends, this is us, my family in New Zealand, my brother and his son. Kent, you're in the audience. This is us in Japan, Tyler also, and Rob. And just finally want to say thanks to my wife too, Yola. It's been amazing. I think I've said many things already, so I won't say that because I might cry. This is Yola just taking a nap with Quincy's dog, Polo. I thought that was really cute. But, thank you. Thank you.
So we have time for some questions from the public, and then we'll have a closed session to finish this off. Any questions from the gallery? All right. Maggie, is this an itch? What are you thinking?
Just have an itch on my nose. That's all.
Oh, there's a question.
Good luck, Kyle. It's been a while. So you measure the NO release kinetics and concentrations in bulk solution,
Yes.
right? So how does that compare with the release kinetics in, a very small environment like a cell? How does that change, and is there a way to measure that?
You mean inside a cell or in?
Yeah, like instead of, so you're doing in bulk solution, how do those kinetics change in the intracellular environment?
Oh, that's a really good question. I think there are some people who have studied that, like this, I think basal levels of nitric oxide, and I think it requires like pretty sophisticated electrochemical setups. I'm not quite sure how the kinetics change, but I wonder, at least, I think at least relatively, if it's releasing fast in solution, it will release faster in inside the cell to compare to, say, like lower release, slow release donor. Yeah. Good question. Anything from the?
Okay, so maybe we'll thank Kyle again, then we'll excuse everyone.